When biologists study the anatomy of ancient plants, they may use a technique called the fossil peel. From a carefully prepared fossil, organic materials can be transferred to a thin sheet of plastic for study under the microscope. This peel shows the cellular structure of a plant stem in cross-section. The plant, Lepidodendron, is a distant relative of the modern club mosses. It grew more than 265 million years ago. This fossil peel, from the same period, shows the cellular structure of ancient fern roots in cross-section. The study of plant fossils helps biologists understand how plants have evolved. The evidence is incomplete, but we can reconstruct the general outline of plant evolution by comparing fossils with living plants. Among the first plants were probably simple algae, like Chlamydomonas. These living fossils contain chlorophyll, enabling them to convert the simple materials that bathe them into food and protoplasm. The earliest land plants may have been primitive algae, like Frichiella from India. To survive on land, plants adapted in several ways to evaporation and other effects of air and sunlight. Most important in the development of all land plants is the epidermis, an outer layer of cells that retards evaporation. Here is the epidermis of a primitive land plant in cross-section, showing one of the openings that permits gas exchange. Through the modification of tissues, land plants have developed specialized organs, leaves. The more leaf area exposed to sunlight, the more food the plant can manufacture through photosynthesis. The plant's main axis, or stem, is another specialized organ. Finally, there is the root system, providing anchorage and absorbing water and minerals from the soil. No one plant developed roots, stem, and leaves all at once. All of these specialized structures depend on the development of a conducting system. A conducting system that permits fluids containing raw materials and manufactured food to flow throughout the plant. These thick-walled conducting cells form what is called the vascular system. All plants with such a vascular system are called tracheophytes, although a vascular system like this is found only in advanced plants, such as the angiosperms. The very ancient plants had less advanced vascular systems. The moss polytricum has a primitive sort of conducting system, although it is classified as non-vascular. In polytricum, this primitive conducting system is composed of elongated living cells. The movement of fluid through such cells would be blocked by the end walls of each cell, and it would be slowed by cytoplasm within the cells. Any flow of fluid would then have to take place by osmosis. If we examine another primitive and ancient plant, one of the tracheophytes, we can see some improvement. The conducting cells of this plant have thin areas in the walls that permit some flow of fluid. Such a plant is Psilotum. Psilotum does not have true leaves or roots. Part of the Psilotum stem grows underground and is called a rhizome. It can absorb water and dissolved minerals through microscopic hair-like processes called rhizoids. From the rhizoids, fluid passes through the outer stem tissues. It is then transported to other parts of the plant. through a primitive conducting system composed of dead cells called the xylem. These xylem cells also give the plant rigidity. Other vascular cells carry the products of photosynthesis and are called phloem. These are living cells. Thus, the vascular system conducts fluids from one remote part to another, and it provides supporting tissues in the plant. Plants that rise above other species can receive more energy from the sun. The club mosses and the horsetails both have small leaves and roots. The evolution of roots and leaves represents an advance among primitive tracheophytes. The ferns are also tracheophytes. They show an even greater advance with the evolution of large, complex leaves. The ferns and their many species occur in abundance. Some gardens and conservatories have ferns from many parts of the world. There are the giant tree ferns of the tropical forests. This fiddlehead is part of a tree fern from Hawaii. The tiny free-floating salvinia lives in water. This delicate maidenhair fern is native to Peru, while the exotic staghorn fern is from Australia. The holly fern grows in Japan. Somewhere on the margin or lower surface of a fern leaf are rows of small dots. These vary according to the species and are called sori. Sori are groups of tiny capsules which contain spores. The spore-producing plant is called a sporophyte. The tiny capsules which make up each sori are called sporangia. Sporangia disperse spores by ejecting them into the air. The mechanism for dispersing spores can be seen in this microscopic view. When the spores are mature, the sporangium bursts and stretches back like a catapult. So that we may see it better, the motion has been slowed down by placing the sporangium in glycerin. Spores are almost microscopic in size. A tiny filament grows out from the spore. Through a process of cell division, this becomes a new plant. Not at all like the familiar fern. This plant is often called a prothallium. On the underside of the prothallium are many rhizoids. Two kinds of reproductive organs are also found here: male sex organs and female sex organs which produce sex cells or gametes. It is called a gametophyte. The male sex organs are called antheridia. Mature sperms leave the antheridia only in the presence of water. They swim by means of flagella. One of these swimming sperms may reach an egg in the female sex organs or archegonia. When a sperm combines with an egg, the fertilized egg or zygote is the first cell of the embryo. This grows through a process of cell division. The new plant, which derives food from the parent gametophyte, is the new sporophyte generation. In time, it grows into the familiar fern plant with well-developed leaves called fronds. A characteristic of fern plants is the way they spread by extending a system of underground stems or rhizomes in many directions. From various parts of the rhizome, grows a network of roots. Ferns, like some of the more primitive plants, include in their life cycle an alternation of generations. A plant of the sporophyte generation will have progeny, often difficult to find in nature, of the gametophyte generation. The gametophyte generation reproduces sexually. From the fertilized egg cell which it produces grows a different generation which reproduces asexually or by means of spores. If we examine other plants, we notice that among the more advanced, the sporophyte generation increases in size and importance. It is in this sporophyte generation that vascular systems have evolved. The alternation of generations can be studied in the laboratory. In a normal gametophyte, the male and female sex cells each contain a specific number of chromosomes. Such cells are said to be haploid. The zygote is formed through the union of a sperm and an egg cell. From this develops a new plant. Each cell contains twice the number of chromosomes and is called diploid. Thus, a normal sporophyte is said to be diploid. Through experimentation, we can study the effect of chromosome number on the alternation of generations. A leaf is removed from a young sporophyte and placed on the culture medium. After a time, most of the cells die, all except for a few cells which begin to grow. These cells develop not into a sporophyte, but into a gametophyte, which is diploid instead of haploid. If self-fertilization occurs, the resulting sporophyte is tetraploid instead of diploid. That is, its cells contain 4 times the number of chromosomes found in normal gametes. If the procedure is repeated with a tetraploid leaf, a gametophyte develops, that is tetraploid. Thus, a simple difference of chromosome number is not what determines the alternation of generations. Other experimental work can be done with ferns. Here, under sterile conditions, a fern plant from the field has been stripped of its outer leaves. What remains are rudimentary leaves, which would normally take 2 or 3 years to mature. When one of these is placed in the culture medium, it shows signs of growth within a week. Chlorophyll develops, and in time elongation takes place along with the formation of a fiddlehead or crozier. After about 6 weeks, the crozier uncurls, revealing a mature leaf or frond. The experimenter can therefore study the growth and development of fern leaves under controlled conditions. A drop of this liquid contains fern spores that have been exposed to intense radiation. The spores are planted in a culture medium. Radiated spores are planted in another tube. This culture medium contains an amino acid, methionine, in low concentration. Each tube is sealed. After 8 weeks, the 2 cultures are compared. The tube without methionine shows gametophytes with a large number of tumors. The culture containing methionine shows almost no tumors. Thus, by inducing tumors in simple ferns, biologists can study life processes which may be related to abnormal growths in animals. In conclusion, we may consider the ferns highly specialized land plants. They have roots, They have stems, shown here in longitudinal section, and they have leaves. All of these specialized organs depend on the evolution of a vascular system. While ferns are evolving, so too are other vascular plants. For a vascular system gives the land plant adaptive advantages. It can spread and grow tall and thereby receive more energy from the sun.